He also wants an introduction.
So I should start, Ronnie? Okay, good afternoon, everyone, and welcome to the colloquium. Actually, I was approached by Ronnie last week on Friday asking me to give a colloquium, and I said yes. I didn't think about it. That's always how I act. Actually, I say yes first and then think, and I find out actually I made a mistake. But one of the things the interesting thing that happened actually is that even though I'm in a computer science department, I don't remember the last time I talked to computer scientists actually. All the talks I give are in biology departments and in biology conferences. I don't talk usually to computer scientists. I don't like them. I like some.
So you don't
like yourself.
You don't consider 182 talking?
No. Actually, 182 students wrote last semester. It's good that it is offered in the spring, because in the fall, students are still under the age of 18, and that would be child abuse. So.
Bravo!
That made my day. So I was asked again to talk, and I asked Ronnie what type of talk, because Anshu had given a talk about graduate school and stuff like that. I thought it was a similar talk, but he said he wanted research. But then I thought about it, again, the issue about research in this area, if I don't talk a lot about the biology to set the ground for it, it's very hard to illustrate. So this is again the first time I talk to computer scientists about this type of work, and I will try to put it in the broader area of data science, because everyone should be a data scientist. And since it's about evolution and survival of the fittest, if you want to survive these days, call yourself a data scientist, you'll be set. Okay? And so I will start with this actually. And this is a quote from Peter Norvig, who's the research director at Google. He said, "We don't have better algorithms, we just have more data." And when someone from Google says something, the community starts debating it, right? Because they listen to Google. And in fact, I was at a very recent meeting on evolution at the University of Michigan in Ann Arbor, and there was a whole session about this, biologists arguing whether they need better algorithms or just more data. Okay. And it's interesting because I went to Google Research and I looked at their website, and they list 808 people, researchers. I don't think they are all collecting data, right? So I don't think even Google buys that statement that they just have more data. They do need better algorithms. But I would even say that the question is missing the point, because people talk about better algorithms or more data, and I have looked, and if you actually search this quote, you will see lots of debates on the internet, and you see people taking one of the two sides. Some people say we need more data, some say we need better algorithms. But to me, what is the question that you are after, right? Because sometimes it depends on the question. It's not the data, it's not the algorithm. In my opinion, the second thing is the model. It's more important than the algorithm, and it's more important than the data. You need to get your model right, or else you are going to get the wrong answer. Efficiently or not efficiently, it doesn't matter, but you're going to get the wrong answer. Then comes the algorithm and the data. And this is what I want to make the theme of this talk, with illustrations from my own research area about the importance of this issue here, the model, and how it fits with the algorithms and data. Okay. So my question, and I put it in double quote because it's not really my question, but this is what I'm interested in, is that given genome-wide sequence data, what is the evolutionary history of the genomes? If you give me genomic data, and I call it genome-wide because it doesn't have to be the whole genome, it can be samples from this genome. Okay, it can be position 1 and position 100 and position million from the genome. I don't have to have the entire genomic sequence. This is what I mean by genome-wide. So why am I interested in this, or why are evolutionary biologists or biologists in general interested in this? It's because if we want to understand how species adapt to their environments, we have to know how they evolve. You hear all the time, actually, about antibiotic resistance, I hope. Actually, there was an article just recently about how bad the meat is in the chain restaurants in the States. And the whole point about that was that all these the beef and chicken is all coming from animals that are fed antibiotics, right? And the whole issue with that is the spread of antibiotic resistance. Antibiotic resistance is evolution in action. Okay, it's evolution in action. It's not something developed. It's not something designed. Nothing like that. It's just something is evolving to become more fit, to adapt to its environment. An environment that has antibiotics, and a bacterium that is not resistant to that is not fit, it's not going to survive. So this is how they adapt to their environment. Evolution of genomes, when we look at these evolutionary trees of genomes, we understand how genes are evolving and how proteins are evolving their functions, how they are acquiring their functions, how they fold, and how they interact and so on. How traits evolve. Why do certain species have certain traits? Or how are traits correlated or not correlated across these traits? So we need all this. We need to understand all these issues. We do need an accurate inference of an evolutionary history of the genomes or of the species. Okay? Now, it's very important to keep in mind that if you want to work in biology, you have to understand that everything that's on Earth that you see today, all kinds of species, are related through evolution. And this is a very famous quote that almost every biologist start their talk by, from Theodosius Dobzhansky. He said, nothing in biology makes sense except in the light of evolution. And this is where in the title it comes in the light of in my slides, and I talk about in the light of population genetics. But what this means, or why Dobzhansky said this, it's based on the Darwin's theory. Okay? One of the main principles of Charles Darwin's theory is descent with modification. That species, we have species and they descend and they diverge. And then some of them go extinct, some of them are still there, but they speciate and diverge and so on. And this is where you get this tree of life that basically captures the evolutionary history of all species on Earth. So this is why you and chimp and gorilla and the oak tree outside are all related, okay, from a common ancestor. Of course, you don't have to believe in that, but that's biology. If you don't, you could have watched the debate last night and heard about vaccines and how they cause autism and all of that. Okay? So before I go into that, I mean, one of the things is that computer scientists, it's not typical in computer science that we would teach something like the concept of likelihood or maximum likelihood in our algorithms courses or our mathematics courses. So before I go and start talking about what it is that we do and what is the modeling, I just wanted to say a few words on models and likelihood, otherwise everything is gibberish after that. Okay? So I just want to make sure that everyone knows what I mean by likelihood, because it's going to show up after this. And if you know what likelihood is, sorry about boring you for the next few minutes, but I want to just talk about this issue. And I'm going to start with a motivational quote that says all models are wrong. I'm going to go after modeling, and we need to understand all models are wrong, but some are useful. This couldn't be more true than in a field like biology. Okay? Because every model I'm going to show and every model that has been derived is for sure wrong. Okay? We are talking about things that evolved over billions of years. Nobody is willing to bet money that this is the right model, right? And it is not going to be the right model, but the important thing is the second part. They are useful, they are supported by the data, and they are resulting in testable hypotheses in biology. So what do I mean here? We are dealing with DNA sequences. A DNA sequence is a finite length string over ACTG. And we will make assumption here. For example, the letters within the string are identically and independently distributed. So if you think about you have a sequence of reads of tossing a coin, and you see heads and tails, that each one of these heads and tails is independent of the ones preceding it or coming after it. This is what we mean by independently. And identically, it means that they are all coming from the same coin, okay? From tossing the same coin. And here are some examples of models. When we talk about a model, we really talk about there are parameters that define that model. Okay? So here's one example of a model. One says that under this model, the probability of ACTG is 0.25. Model 2 is the probability of A and T is 0.1, C and G is 0.4. This is actually realistic in many areas in many areas of the genome. There are regions in the genome that are rich in G's and C's. Okay? This is a model that says ACTG have very low probability, ACT have a low probability, and G has a higher probability. So these are three possible models, and I can list an infinite number of them, right? Because I can keep listing different probabilities. So this is what we mean by models. This is as simple as this here, and we leave it at that. When we talk about the likelihood is think about it as a measure of the goodness of a model. When I talk about the likelihood of a model, I'm really interested in saying, well, how good is this model given the data? So we now need to start talking about the data as well. The likelihood of a model, which is denoted by theta, given the data, is nothing but the probability of the data given that model. So what is the probability that this model could have generated this data? Okay. So if we go back to these three models here, and I take this data, so I need to look at the data. So here's the data that was generated by some model that's generating letters one at a time, identically and independently distributed. If this is a sequence, I can ask you now, what is the probability of this data under this model, or what is the likelihood of this model given this data? Okay? And it's trivial, actually. The probability of this data given this model is 0.25 to the fifth. 0.25 times 0.25, 0.2. The assumption of independence is allowing me to multiply these probabilities. The assumption of independently, identically distributed is allowing me to use the same model for all of them. And I can quantify the likelihood of each one of these three models. Okay? This is really what the likelihood is. What is the probability that this data came from this model? Of course, we are interested in finding these parameters, right? So the interesting problem usually is that you have data, but you need to figure out the model. Sequence data, there are lots of it. Actually, you don't have to be biologists, you don't have to have Will wet lab to have access to sequences. There are billions of nucleotides that are stored in public databases. You can go and access them. But the question of interest is, can we find the model? One way to define the model is actually to go after this optimality criterion, which is, find me the model that will maximize the likelihood. So of all possible models, given this data, which model has the highest likelihood? Okay? Make sense? So if I go back to the previous example here are the space of the models that I have. I have three of them. I need to compute the likelihood, and I need to find the one that maximizes that, and it turns out it's this one. Okay? So this one here is the one that has. So under maximum likelihood, the maximum likelihood estimate of the model is model 2. Okay? Fine. That's all I'm going to say about models and likelihood, because I'm going to be talking about likelihood and maximum likelihood estimates. Okay. So now back to evolution. We don't have coins or anything like that. Yes?
I have a question. So my biology is a bit tricky, but if I remember well, then three genome makes an amino acid, right?
Mm-hmm.
So why don't you lift the model up to the amino acid level and then look for likelihood instead of in the letter?
So the question is, why are we working with DNA, not with amino acids?
Yeah.
Yeah, you define models for what? For DNA, we have models for amino acids, we have models for RNA, we have models for all these things. And even for DNA, it doesn't have to be independently distributed. Like the independence part, we can also relax it and or remove it and put some Markovian dependency and so on. Yeah, I mean, it's much more complex than this in biology, but I was just illustrating a simple model. But in general, it's not, yes, protein is more conserved than DNA, but it's not always the case that you want to use protein when you construct trees. Because also protein have very much lower evolutionary rate, because proteins have to be conserved, because they perform some function. So especially for closely related species, you don't want to use protein sequences. But that starts getting into the biology of it. Okay. So now that we know what is the model, what is likelihood, so I'm going back to evolution, what is that model that we are after, and how do we find it? Okay. So the data, in this case, is not a sequence of letters that were generated by some toss of a coin or a four-sided die. We have 4 sequences here that are coming from different species or different individuals here. These could be 4 sequences coming from 4 individuals sitting in the room, or from human, chimp, gorilla, mouse. Okay? And this is the data. The data, I'm going to assume that it is what we call aligned, that they are all of the same length. Okay? When biologists deal with the data, it's never like this. So there is a step before this that's called alignment. And we have to solve that problem before you do this. If you want to learn that, take 182. I feel bad for the students. So what is the question that we are interested in once we have that data that's aligned? What is the evolutionary tree of these sequences? Okay. So to understand that, or to figure out how to find a model now, again, the model is no longer that simple. You take a coin and toss it, or take a die and roll it, and list heads and tails and so on. Now you think about these 4 sequences that I just showed on the previous slide. They are coming actually from a generative model that looks like this. What is this generative model? It is a rooted binary tree. There was a sequence at the root. This sequence started evolving down these red lines, or the edges of the tree. these lines have lengths. Okay, so this is shorter than this. These lengths correspond to mutation rate. Okay, so this here we had fewer mutations that happen, or we expect fewer mutations to happen here than to happen here. So this sequence mutated to this one. This sequence now mutated to this and to this. This sequence mutated here and then this one. Now the issue is, in evolution, when you're looking at evolutionary data or biological data in general, the only thing you have access to is the blue parts. These are the sequences that we can have access to today. You can take any of you interested in figuring out your genome, you can actually go today and give samples and they will give you your genome sequence, okay, on a USB drive, and you can then look at it. But the thing here is that when you have these sequences and I'm trying to build this tree, I don't have access to any of this. I don't know what these sequences were, I don't know the length of these edges, and I don't know the topology of the tree either. So the model now, again, is not that thing that we toss and has 4 sides or 4 possibilities. The model now is a rooted binary tree whose leaves are bijectively labeled by the sequences. So there's a unique labeling. Okay, it's 1-to-1 and on-to. And we have branch lengths. So the model, I have to figure out, if I want to find the phylogenetic tree, I have to figure out the rooted binary tree, and I have to figure out the branch lengths. Notice that I'm not saying anything about the sequences that were on the previous slide. I don't want the green sequences. Okay? You will see the formulation now that we will actually say, I don't know what they are, I'm going to sum over all possibilities. Okay, which differs from if you have taken machine learning and seen these models like Bayesian networks and so on, this is very different. Okay? And especially when we go to the non-tree model. Okay, so the inference is that I have, this is the data that we have, and this is the thing that we are interested in. I'm interested in the topology, how things branched and diverged from each other, as well as the length of these branches. So this is the likelihood under that model. So this is the probability of the sequences given the gene tree or the model. This product here is taken over all the sites. I'm assuming the sites are independent, so I can take the product over the probabilities of these sites. Then this summation, as I said in the previous slide, even though there are these green sequences that were at ancestral nodes, we don't know what they are, we are going to sum over all these possibilities. And then we are going to take the product over all branches or over all edges of the tree, and then the probability of the sequence at one end of the branch turning into another one. Okay? So this is this probability here is still saying for a branch whose tail is U and whose head is V, from U to V, what is the probability that the sequence labeling U becomes V after the time that's given by that branch length? Okay? So that we need to incorporate the branch length, and something that's not shown here is known as the model of evolution. Okay? So there are different models of sequence evolution. And this, again, this summation here is basically saying, I don't know what sequences were at the ancestral nodes, I'm going to sum them, and basically, the term to use is here, something like integrating them out. I don't know what they are, I'm going to integrate over all possible sequences. They are discrete, so this is why it's summation. And this is the MLE here, the maximum likelihood estimate. Under this model here, now, what is the tree G with its branch lengths that's going to give me the highest likelihood? Okay.
How long is that model dates back?
This one here?
Yeah.
This model was actually introduced in the late 70s and the early 80s. Yeah. It was introduced by a very famous population geneticist. His name is Joe Felsenstein, who's now at the University of Washington. And one of the interesting things that he introduced this, and he wrote the paper on maximum likelihood. The paper today has about 8,000 citations. And one of the interesting things is that in the first 10 years when the paper was introduced, it had only 60 citations. And then it exploded once the genome sequencing actually came on the scene, because now you had data to be able to apply statistical models. Before that, actually in his paper, I think he applied things even manually. He was computing things like manually. So this is the likelihood. This is the maximum likelihood problem. This is computable in polynomial time. It's known actually Felsenstein's algorithm. It's a very elegant, beautiful algorithm. It's dynamic programming on trees. You go from the leaves of the tree towards the root, and when you are going, you compute these probabilities and store them at internal nodes and make use of them. Okay? It's known as Felsenstein's algorithm. And no surprise, this is NP-hard. Okay? So if you give me the tree, given by its topology and branch lengths, I can compute the likelihood very easily. If you give me just the sequence and say, find me the tree, that's NP hard. Okay? And as whenever we have NP hard problem, of course, one of the solutions is heuristics. So now you can do hill climbing techniques, whatever you want, whatever these search techniques. You can start walking in the space of trees. Every time you see a candidate, you compute its likelihood very quickly, and you can continue with that. So that's maximum likelihood. And I want to go back to the issue of better algorithms for more data. Here's a very simple phylogenetic tree. It has 4 taxa. It's shown as unrooted. Okay? The tree is unrooted. What happens in this tree is that we have these A, B, we have their long branches here. C and D have very short branches. What I am showing you here, how maximum likelihood would perform as you increase the amount of data. So this is, for example, here, imagine that you are giving me 4 sequences from A, B, C, D of length 1, here, of length 2, 3, 4, and keep increasing the length. And here's a different model that is also very popular in phylogenetics. It's called maximum parsimony. This is what happens in phylogenetics when you feed the method more data. This is, we are increasing amount of data. It's the same data I'm feeding it to the same method. Both are using the same algorithm, hill climbing. One of them converges to accuracy 1, the true tree. One of them converges with high confidence to the wrong tree. This is another example of, it's not about the data. You give maximum parsimony more data, it's going to converge, and it's going to give you an answer with very high confidence that is the wrong answer. Okay? And this is maximum likelihood. They both use hill climbing. They are both using the same data. It's the same algorithm, the same data. The model is different. Okay? So that's basically what phylogenetics, and again, this was done in the early 80s by Felsenstein. This is not the state of the art. I mean, the field has moved also into Bayesian phylogenetics, where you don't go after an MLE estimate. You go actually after sampling the posterior of phylogenetic trees and so on. But I want to move now to the post-genomic data, which is actually much more complex. And this is where we are doing some of the work there. So what happens in the post-genomic era? Again, before we had genomes, it's basically the biologist has interest in 5 individuals or 5 species or whatever. They went and sequenced one genome. A locus in biology means a position in the genome. Okay? This locus basically says here's the sequence or the region that you are sequencing in that genome. But what happens after the sequencing technologies have improved is that biologists easily have access to this data, where from these 5 species or 5 individuals or whatever they are, they have data on multiple genes or multiple regions in the genome. Okay? Now, what the interesting thing that's happening is that if you take every one of these and build the tree using, for example, maximum likelihood, implement that maximum likelihood technique and build a tree on each one, this is the scenario they're going to get. And if you look at these things carefully, you're going to start seeing that the trees are different. For example, this one here says A, B, then C, then D, E. This one here says B is closer to C. If you look at this B, C, and this is B, E. Okay? And the same thing here, now C all of a sudden closer to D, and this. Now, each one of these is a fine tree for the gene that we built it from. Okay? But the question is, how did the genome evolve? Is it this? Is it this? Is it this? Or is it none of them? Okay? So that's the question that the community is interested in now, because one gene is not going to tell me how the species evolved or how they adapt to their environment. I need to understand how the entire genome evolved, and so that I understand what are the events that operated on that genome while it was evolving. Okay? So the interesting question is to go from this to this genome phylogeny or evolutionary history of the genome. So here's a story from a Nature paper that appeared a few years ago. As you can see, biologists like each other and they collaborate. Unlike computer scientists. But we are still much better than mathematicians. So this is a paper that looked actually at the evolution of human, chimp, and gorilla genomes. Okay? And one of the interesting observations they found was, so they had the whole sequences of these genomes. One of the observations is that the current understanding or the hypothesis about how these species evolved is that humans are closer to chimp than chimp to gorilla. It might not sit well with some of you, but you are closer to chimp than chimp is closer to gorilla. Okay? Hopefully you're not offended. But one of the interesting things here again, in 30% of the genome, if you look at 30% of the genome, not one contiguous region, but across the genome, 30% of the genome is actually point to a different story. 30% of the, about 15% tells us that we are closer to gorilla. So what that means that if you're a biologist and sequenced a region from that part, the tree is going to tell you human is closer to gorilla. Another 15% is going to say chimp is closer to gorilla. Okay? So 70% is putting chimp closer to human, 15% putting chimp closer to gorilla, 15% is putting human closer to gorilla. Okay? And this is a very famous phenomenon now. There's a lot of interest in it in biology because we are starting, or biologists starting to sequence very closely related species in multiple individuals. This is known as incomplete lineage sorting. Okay, this is the phenomenon known in biology as incomplete lineage sorting. So this is the scenario now. Again, these are just 3 species. There are 3 possible topologies on 3 taxa actually. If you think about how many binary trees there are on ABC, there are 3 possibilities. And all of them show up with these percentages. Okay? So the question now becomes, what is the evolutionary history of the genomes, given that individual regions are giving us conflicting signals? One is saying human closer to chimp, one chimp is closer to gorilla, and so on. What is it? What is the true evolutionary history? Why do we think that human and chimp are closer to each other? To answer that question, we have to understand why do these trees disagree. This is where you have to stop sitting in Duncan Hall and go talk to biologists. Right? This is not something that let's play with math. This is something that you have to start understanding biology. Okay? And to understand biology, this is where the lens of population genetics comes into the picture. We need to ask now, how could this gene tree have arisen given this by chance? Okay, so here's the same story, human, this is Darwin, chimp, I don't know what's his name, or this is gorilla. So we have now this evolutionary history of species, and what I'm drawing inside each species here, you'll see that there are multiple blue dots. These blue dots are individuals. Okay, so think about this as every row here showing you the individuals in that population. Like if there's a human, imagine that we have a few billion dots there, one for each individual. And this is whatever number of chimp individuals, whatever number of gorilla individuals. Now think about the following. You take a copy from human. I take the gene copy from my genome, the gene copy from a chimp genome, and a gene copy from the gorilla genome. And now I want to trace their evolution backward in time until they meet at one point. All 3 of them, they coalesce into one point. This is known as the coalescence. So you start going back in time until you get to this split point here. Nothing interesting can happen with that point because you are just following within the gorilla population or the chimp population and so on. But where things can start getting interesting is that in this branch here. So now we have entered, when we are going backward in time, we have entered that ancestral population where neither human nor chimp existed. There was an ancestral species there. And this is the species that was there. Now these red lines start thinking about how they are going to trace. And in every generation, just ask a simple question. What is the probability that, I'm at a certain generation now and I take these 2 red points here and they meet in the previous generation? Okay, so imagine this question in probability that you have n points and you have another row of n points, and I take 2 points from here and I say, what is the probability that they meet at the previous generation? If there are n points, the probability is 1 over n. For them not to meet is 1 minus 1 over n. Okay? So if I want to get this tree, human and chimp, these 2 genes that I chose, they cannot meet. They cannot meet here. If they met, I cannot just get this tree anymore because I'm putting human with chimp already. So for this to happen, there are 2 conditions need to happen. One is that human and chimp need to keep missing each other here. They should not hit the same point. Then when we enter this point, there are 3 lineages or 3 red points. There are either gonna merge first the human and chimp, or the human and gorilla, or chimp and gorilla. We want the right one from them. Okay? And we can derive the probability actually. The probability that these 2 do not coalesce here is 1 minus 1 over n to the t2 minus t1. This is the length of this, or the number of rows in this branch here. And the probability now that once they enter here that they coalesce, there's a main assumption here that they are given infinite amount of time for that to happen. So we give them infinite amount of time that they will coalesce into one point. The probability here is 1/3. There is no difference because there are tons of assumptions under this model. So the probability now of this tree, given this is the probability that this happens. So we can now derive this model of what is the probability of a gene tree, and we can try to explain why is it happening like that with human, chimp, gorilla. And these are actually the three probabilities. That the probability that a gene will tell me human is closer to chimp is 1 minus 2/3 is 1. And the probability that human goes with gorilla or chimp goes with gorilla is equal. Okay. And here's I'm illustrating these three probabilities as a function of this variable here, t2 minus t1 over n. The longer this is, or the bigger this is, you see that the human, chimp, gorilla, the human and chimp go together with higher probability. Okay? So what this means is that the longer this branch or the narrower it is, the higher the probability that if you sample a gene by randomly from the genome, it's going to give you the right evolutionary history of the genome. Okay? There was a question?
Is there a possibility of the cycles in this graph?
Is there a possibility of what?
Is there a possibility of cycles in the graph?
Cycles.
Yeah.
Okay. That's going to lead us to some promiscuity and stuff, interesting stuff there. What does it mean to have a cycle? This is whole, I mean, you're talking about evolution and sexual reproduction. Now figure it out. What happens? This talk is rated PG-13. No, cycles, we don't have cycles in evolution. You can have, in the underlying undirected graph, you can have a cycle, but in a directed fashion, to have a cycle, no. So now again, as this quantity becomes larger, the probability of the gene tree that agrees with the species tree becomes higher. So if I show you this, do you think you have an algorithm? Suppose a biologist has 3 genomes and want to understand or to figure out what is the evolutionary history of how the species evolved. Do you have an algorithm based on this? Suppose we are operating here. Do you have an algorithm for the biologist to say, do this, and you will get the species tree? So I can tell the biologist, go build gene trees on, like, take thousands of genes and build gene trees on them. Then what? Look at the one with the highest frequency. Because this is saying that the gene tree with the highest frequency is going to agree with that, right? So what this means here, if you are in this region here and you sequence 3,000 genes and you build their trees, what this is saying is that a large majority of these trees are going to agree. So just sequence them, build the trees, and look at that tree with the highest frequency.
So a few minutes ago you said something about at some point you need to go and actually talk to the biologist about why this is happening. You can't just use math anymore. So in this example we have these 2 divergent subtrees of the same superspecies of human and chimp that didn't ever cross over. It seems like things like that could happen all the time in nature when populations get divided. But it seems like the mathematical models we're using here are simply like, what are the probabilities that 2 members of the subspecies never reproduced or something like that without any of these external factors. So
Like human and chimp reproducing?
No, 2 members of the super species.
Super ancestral species.
The ancestral species, yeah.
That,
So where do the external factors come into play here? Do you just, since we can't figure out the external factors since they happened a long time ago, you just use the math, or?
remember that all models are wrong and some are useful. And when you do modeling, you are abstracting so much. There are lots of assumptions. Actually, the point you're raising is very important. There are lots of assumptions. This model, deriving these probabilities, is done under a model known as the Wright-Fisher model in population genetics. It was derived in 1915, something like that. And there are lots of assumptions there, like random mating and no population structure and no gene flow and so on. But under these assumptions, we can do this. Now, when you're talking about 2 individuals within this species, can they mate and can they reproduce? The answer is yes. It's not going to violate this model. Because what I'm showing you here, again, I abstracted it. I said everything is an individual, but every point here is really 1 gene from 1 individual. I'm not talking about multiple genes from there. But I'll be happy to answer this after the talk if you still have questions about that. And the second thing is that very soon I'm going to actually talk to a point where it's not just incomplete linear solving, something else can complicate this picture. Okay. So based on this, again, if we develop this model and we look at this, then an algorithm naturally comes, which is this: sequence large number of loci, build their gene trees, and return the most frequent gene tree. Right? Should work. Actually, asymptotically, you can easily show that as the number of genes goes to infinity, you'll get the true species tree with probability 1. But it turns out actually it's more complicated than this. And there was this seminal paper that appeared actually in 2009, I believe, that showed mathematically that you can actually find settings for that species tree, that human chimp gorilla. You can put actually links on these branches to produce a very interesting case that the most probable gene tree to appear actually is the one that does not agree with that species tree. This was a big result actually when it came out a few years ago. Okay, we are talking about, here's a model, generate data under there. The data is finite, I mean there are finite possibilities. Generate an infinite number of objects. Look at the one that appears with the highest frequency. It turns out it's not the same as does not reflect the true model. So this is another case where the more data, the higher confidence in a wrong answer. Okay? Give it more data to that method, which is known as the majority vote. And keep just adding more and more data, you are going to keep converging. You will converge to the wrong answer and you're getting higher confidence in that. Okay? So now we have to do a modeling a bit better and think about as the data being the sequences. So we have now remember I talked about multiple loci or multiple regions in the genome. We have this sequence alignment for the first region, for another sequence alignment, an M sequence alignment. And I'm interested now in the genome or species tree side. It's the topology and the branch lengths. This is the likelihood of the model. Okay. So the likelihood of the tree given the sequences, I have this product here that denotes, I'm assuming the loci or the regions are independent. So I can compute the likelihood of the tree based on each one of them independently and take the product. Similar to before where we summed over the ancestral sequences, now I can integrate over all gene trees. Gene tree topology is a discrete object and there's a finite number of them. We could have done this as a summation, but remember that these gene trees come with branch lengths. So now we are summing over all gene tree topologies, but we are integrating over all possible branch lengths. So this is what makes it infinite, and we have this integral over it. And what this is saying here, that now the generative model of the data, when you observe these genomic sequences, there is really a nested model here. There's that big tree of tubes, the species tree, that generates these gene trees inside, under that model that I showed with the probabilities of 1/3, e to the minus whatever. And then on that gene tree, there's a mutation working that generates the sequences. Okay? So to generate genomic data, you can first generate a set of gene trees from the species tree under that model, what we call the coalescent. Then for every gene tree, with its branch lengths and a model of sequence evolution, it defines a generative model that you can generate sequences from. Okay? And this becomes the likelihood here. And of course, if we have just the sequences that was talking before in the pre-genomic era, this is what we were dealing with. There was no species tree, gene tree. We were assuming everything coming from one tree, and we were trying to find the tree that maximizes this. Now it's a bit more complicated because each one of them could have come down any of the gene trees, and each of the gene tree could have come under that species tree. Okay? This is what was computable using Fosenstein's algorithm, and it's still appearing here. Now, there's one of the simplification techniques that are done in practice. Instead of going from the sequences directly, biologists usually break it down into two phases. The first one, they take the sequences and build gene trees. And then the second one, we've tried to find the species tree that reconciles or gets this species tree under the maximum margin. Okay? So now, to get from the sequences, from individual sequences to individual sequence alignments to gene trees, it's done. It's been done for decades now. You can run any of the methods out there that gives you a phylogenetic tree. But to get now from these gene trees to that species tree, now this is a likelihood model, and this leaves us with this question: what is the probability of a gene tree given a species tree? And I have shown it already. This is the case for 3 taxa. Okay, so if we have 3 species, this is the probability. So you can take this times Felsenstein's algorithm will give you the whole likelihood. But this is for 3 taxa, and this is how it gets when you have more than 3 taxa. Okay? It has this nasty function inside. This tells us what is the probability of a certain number of lines entering the branch and a certain number exiting that branch. It's actually a mess to compute. But then we have this summation over all possible ways that gene could have arisen from that species tree and these combinatorial terms. The point is that we know how to compute that probability. Okay? And in this case now, this problem is conjectured to be NP hard. Getting the species tree under that likelihood formulation is conjectured to be NP hard. I don't think anyone is going to be shocked if someone proves it to be NP hard. Actually, everyone thinks it is NP hard. And local search heuristics have been developed and devised and used. Okay? So now the last part, let's go back to the question, and again it's going to touch on the same question that I was asked here. Why do individual loci give conflicting signals? I was describing a simple model. Here are 2 points. You go back in time. What is the probability that they coalesce, and so on. That's incomplete lineage sorting, and it is a major factor in evolutionary biology and the evolution of species, but it's much more complex than that. It's much more complex, and here are some of the known knowns in biology. Okay? This is incomplete lineage sorting. I just described it where things go back and miss each other and start coalescing in a different way. There's something else that happens in biology known as gene duplication. A gene physically gets duplicated, so now you have 2 copies of that gene, 2 copies of the sequence in the genome, and then they start evolving and they can duplicate more. And this is actually a very important process that there's a lot of interest in it, for example, in cancer biology and in medicine, because if you have ever heard of copy number variation, where there are variations in the number of copies of a gene in different genome, it's because of gene duplication. Okay? And there is another thing, which is again mating between species, between different species. It happens a lot in plants. Okay? Lots of hybridization happens in plants. Again, as I tell sometimes in my course, even if you go to the grocery store, some of the fruits you buy there are hybrids. Okay? Plum cuts and stuff like that. Yeah. They are fine, safe for you, don't worry. Another thing that happens actually under hybridization is horizontal gene transfer in bacteria. Again, this is one of the major examples of this kind of event where bacteria, they are not just evolving vertically from ancestor to descendant, but every now and then, bacteria come together actually physically, the cells combine and exchange DNA. And when that exchange happens, if the exchange is carrying some gene that is responsible for resistance to antibiotics and so on, this is how they acquire it. Okay? And there's a big question in biology about what is driving evolution. Is it point mutations where A goes to C and to G, or is it something like this? Especially in the area of bacteria, lots of biologists believe that this is the main mode of evolution. Things are happening horizontally like this. Okay? So one of the recent stories actually that happened, in bacteria we always hear about antibiotic resistance, but this is a big story that also was published in Science a few months ago, where the authors focused on malaria, and they looked at the mosquito species that carry this plasmodium. And the interesting thing for the main biological question of interest to them was, why is it that these mosquitoes can carry malaria and humans die, right? Again, this is adaptation to the environment and adaptation to the parasite, because they can carry it, but humans don't actually do well with it. And they did sequence the genomes, and they looked at the gene trees, and they did all sorts of analysis. But the point that came from here is that these mosquitoes actually are not evolving down a clean tree. Okay? That this is actually an article written by Andrew Clark and Philip Messer. Clark is one of the big names in population genetics today. And basically one of the things that we said, we need to dispense probably with the tree and acknowledge that these genomes are best described by a network. So the evolution of these mosquitoes is not really a tree, it's probably this type of one. That doesn't mean, by the way, Darwin was wrong, okay? But just for the record people, okay? And then they continue and say there are 2 possible reasons of why we are seeing all these gene trees disagreeing with each other and why this evolutionary history is so jumbled there. One of them is introgression. For now, just take it as hybridization, that this sharing across species. And the other one is incomplete lineage sorting, which is just the chance, okay? The effect of chance. So now this raises the question is that it could be this, and it could be a combination of the 2. How do we do it? Now it's getting more complicated. It's not just I can trace these points back in time and compute these probabilities. I have also to account for the fact that I'm going back, these hybridization events could have happened. So this is a scenario here where you have this species that is part of its genetic material is coming from the left, part of the genetic material coming from the right, and this is what we mean by this scenario here. Now the model changes. We don't have a tree anymore. We have a DAG, a rooted DAG, rooted directed acyclic graph. And the same thing as before, we have branch lengths here. But also we have, in addition to these branch lengths, we have certain probabilities associated with these nodes of in degree 2 that tell us if I'm going backward in time, and you had now a node of the in degree 2, you have to know where to go left or right. These probabilities are going to tell you that. We didn't need it in a tree because when you're going from the leaves to the root in a tree, there's always a single path. But here, when you do that in a DAG, you can hit a node with two parents, and you need to know which one to trace. And this is where these probabilities come into the picture. Yeah.
How often do you think this hybridization occurs? Is it mostly occurring with just local patches of GAG or more serious than that?
In bacteria, actually, it's very complex like this. In bacteria, it's very complex. In plants, this happens a lot, and there is a lot of them. The interesting question for biologists in other types of eukaryotes, especially primates and so on, is how often does it happen? And it becomes like a circular question, because now they are using tools, they are using a tree building tool to try to detect this hybridization, and then they say, look, I built a tree, there's no hybridization. And my argument to them is that you got a tree because you used a tool that returns only trees. Right? So the proof that you have from the data is that the method that you used really builds trees, nothing else. So the question is really circular there because the tools are not yet there to be able to investigate this. I mean, they are being developed now, and they are being applied. Mosquito, now there's no question about it that they have been hybridizing. Mice have been hybridizing, and wheat and yeast and all of that have been hybridizing. So I think with the development of the right models and the right algorithms, I think the extent of this is going to be revealed and is going to be much larger than biologists have thought. So now we have, again, this model. The data is the same as before. We have these sequence alignments. And what we are interested in, similar to before, the only difference now is that instead of this side being a tree, it is a DAG now. Okay? And the same thing as before, if we have the gene trees have been built, I don't have to go from sequences, I don't have to integrate out anything. And the probabilities now become a bit more complicated because when you go in this DAG, the number of paths and accounting for these probabilities of going left and right, you have to take that into account. If you incorporate branch lengths on the gene trees, it gets even nastier in terms of the probabilities, but we know how to compute that. Okay? And again, this is conjectured to be NP hard. If it's NP hard to build a tree, it's NP hard to build a DAG. And local search heuristics have been developed as well for this. I want to finish with one analysis, another illustration of the issue of models and algorithms that differs from the ones before. Before was maximum likelihood versus parsimony. Both of them are returning tree, but they want to return the wrong tree. Okay? And then we talked about the issue of the probability of the gene tree. The most probable gene tree might disagree with the species tree. No matter what, you get a tree, but it's a wrong tree. But here's a case where this yeast data set was analyzed by a tree-based method. And they got an answer with 100% bootstrap support, 100% confidence in this tree. And this tree, same tree, was returned by a Bayesian method that was run for 400 hours on the data set to return this result. And it was analyzed by a parsimony method also for a few minutes, returned the same tree. All of these returned the same tree, all of them returned the same confidence. But all of them are accounting just for that chance factor. Okay? Now what happened when we accounted for hybridization is that you start seeing these hybridization events. Okay? So this takes me back again to that question of more better algorithms or more data. This is a case where if your algorithm, if your model is not accounting for an event, even if you feed it more data, you are not going to get that event. Okay. So to summarize, there's an intricate relationship between gene and genome evolution. It's not a simple thing. Actually, these three events I showed here are, three main events in biology, but actually in practice, the biggest cause of gene tree disagreeing with each other is the errors in the method that built the gene trees. So you will almost never get a correct gene tree, 100% correct gene tree. So now you are dealing with data that already has error in it from other methods that have built that. Okay? Because our the original data is sequences, but then we build gene trees from them. There's a lot of steps there that can cause lots of mistakes and problems with the gene trees. The sequence alignment can be wrong. The phylogenetic tree that we inferred for individual genes could be wrong. And that adds another major source of headache to this inference. But the relationship itself, even with, if we assume the gene trees are correct, biologically it's very complicated. Statistical modeling provides a powerful tool in this area. It's impossible to do this kind of analysis without statistical modeling and inference. You know, combinatorial optimization and combinatorial modeling can take you so far in this area. You have to start thinking about these questions and what is the probability of things happening. Efficient algorithms and big data are good, but proper models are essential, at least in this area. Computational complexity is a major bottleneck. I don't want to take from this that, oh, just get the model right, the algorithms are going to figure themselves out. All these are NP hard problems. You do hill climbing, you are always stuck on these local optima, and it takes forever. The tree and network space is enormous, even if you forget about branch lengths. Okay, like even just with 30 leaves in these trees, the number of possible trees and networks is a number with 20 digits in it. Okay, so this is not something that you can do brute force on or hope for the best by just running a hill climb on it. And while big data often delivers a better signal to noise ratio, add more data, probably get more signal. It's very important to keep in mind that also adding more data adds more complexity to the model. If a biologist focuses on one gene, it is fine probably for the biologist to say, I will rule out hybridization and duplication and so on. But it's not easy for the biologist to keep making that assumption as you start including more and more and more, because the chance of these things happening in other parts starts increasing. Okay? So you want to add more data so that you have more signal. But when you add more data, you have to be careful, because now any assumptions on the model you make can be easily violated because of the sheer size of the data. And with that, I want to acknowledge all the group members. I'm not going to read their names, the former and current, many undergrads who work with me, and many collaborators, the funding agencies. And with that, I thank you very much.
You started off by saying that all models are wrong, but some are useful. But in what way are these useful?
That they are giving evolutionary histories and inferences about the genomes and the gene evolution and how things have adapted that have been testable. Like again, when we talk about antibiotic resistance, for example, it has been shown that when they build these gene trees and map mutations on these trees, and they did experiments in the lab, they are seeing these effects. For example, here in cell biology, just in the next building, next door, Yusuf Shamu actually does what is known as experimental evolution. He actually evolves bacteria in the lab, and he subjects them to antibiotics. And he sequences them every certain period of time. You can do that with bacteria because they have very short generation time. And he can actually now sequence these genomes, actually can look at the mutations and can test these hypotheses and the stories that come from these.
I mean, just a quick comment on your whole Google thing. I mean, I think a fundamental difference is that in the Google world, right, they're mostly talking about supervised learning, right, where you have you have the answer. And I would assume that except in extremely limited cases like you just described, you don't have the answer, right? I mean, God is not sitting there, or nobody is sitting there telling you what the evolutionary history is, right?
There were 11 people on the stage last night who could tell. But the quote from, if I'm not mistaken, the quote from Google always comes in the context of examples on spell checking and word segmentation and part of speech and stuff like that. But even there, I would disagree that it is just the algorithm, because they still have a model and they still have an assumption. And again, that's another thing we give in 182 now, in part of speech tagging, where you have to assume what are these states of the hidden Markov models and so on. So I agree in some sense when you have tasks where you have large amounts of data and you're trying to find patterns, I think the amount of data there is very, very useful. But what I was trying to show here is that again, how common is hybridization and so on. If you don't start by acknowledging that it could have happened and incorporated it in the model, it's not easy. I don't see how the data is going to ever. The data might, one thing that the data could tell me is that the tree model is violated and there is something else happening. But without incorporating this stuff in the model, I don't see how to find it out. But again, I mean, I'm sure there are many tasks that like one of the cases they make is that it's not easy to keep a dictionary up to date and so on. So now you can have millions of texts and megabytes of documents that you can learn the English language from, or the vocabulary in the English language. I agree in that area, right? Because again, you can aggregate as much data and you can find the words and the relationships among the words, subject and noun and verb and so on. But in many areas, I think one has to, the toughest step there is getting the model right.
So you talked about maximum parsimony and peak giving it the wrong answer with more data. Is it because the algorithm is wrong? With more data it should, is it the problem in the algorithm or something more?
No, the parsimony criterion itself.
Okay.
Even if we could solve that problem exactly, it is in pretty hard. Even if you could solve that problem exactly, and you can solve it exactly, it's for text there. If you look in brute force manner at all trees, and compute the parsimony, which is easy to compute on a specific tree, you will get that problem. The parsimony criterion suffers under that tree that I showed. This is a case known as the long branch attraction problem for parsimony. It does happen in biology. It has been shown that biological data can have that problem in them. Yeah. So it's not the algorithm. Yeah.
One of the classical definitions of species in biology has to do with whether two individuals can reproduce with each other. That is, if they cannot reproduce with each other, then they are different species. So for the case of hybridization, say in eukaryotes, if they could hybridize, that would mean that they could produce offspring, correct? So couldn't you just treat them as being part of the same species and then introduce to a non-hybridized problem?
Yeah, it's a very good question, and biologists are arguing and have been arguing for decades and centuries about what is a species, what is the concept of species, and actually at the meeting in the University of Michigan, there was a talk by a biologist from England. He's a very famous biologist, James Mallet, who was talking about hybridization, and the title of his talk was, due to hybridization, is WTF species. Okay, I'm not going to spell it out. But this was the title of his talk. What is species when we talk about hybridization? That's one definition of species, which is known as the biological concept of species, where two things are considered species if they are reproductively isolated. They cannot mate. Then if we talk about them as two different species and somehow they mate and create something, then how are they species? But these are different definitions of the species. But this is definitely a very thorny issue. I would say it's outside computer science. And there are lots of interesting articles and opinions, and biologists disagree on it. It's not that they have agreement on the notion of what is species. This is why in certain places I was trying to call it genome tree. Okay? Because once you say species tree, especially when you go to bacteria, what is species there? Right? I mean, why are these two considered E. coli and these two are considered E. coli and Salmonella? It's very hard there. But yeah, it's a very good question.
I like Dr. Goldman's question. I'd like to ask it in a different way to hopefully get a wider response. Doctor, in your opinion...
Since when you call me doctor?
Trying to stay polite for the crowd. But doctor, in your opinion, why is it important for our civilization to have an accurate understanding of the tree or network of life?
Actually, this is something very important for me personally, and I usually don't talk about it when we are talking about science, but I can tell you, and I try to stay away from it. I'm not the most politically correct person or the most diplomatic person, and in the Middle East we are not usually diplomatic. This is why we have peace. But I was born to a Catholic Palestinian family in Israel, and I lived in the Holy Land that has Christianity and Islam and Judaism, and it is the center for them in some sense. And I have seen firsthand the hatred that they have to each other because of religion. Again, people disagree with me, but I'm just going to give you my answer. The hatred of religion, and I think religion is a serious problem, and I think that having the tree of life and understanding how species evolve and communicating that to the general public so that they understand what evolution is. No biologist ever said that humans came from monkeys. That's what intelligent design people say, so that's what repel people about it. But if we understand that and if we educate the public, the scientific data is overwhelming for that out there. And I think it's the sooner we educate the public about this and the more we show them this notion of tree of life, the faster we bring about the death of that idea of some invisible man sitting in the sky, deciding things, and millions of people are killing each other in his name. I think that is, for me, that's more important than every research publication, and more important than tenure and grants. Okay? Because I think at the end of the day, just look around you what's happening. Look at the hundreds of thousands of refugees that crossing the borders and look at what's happening. It's all in the name of religion. And to get rid of religion, we need to understand this beautiful theory of the greatest scientist ever, Charles Darwin. The problem with it today is that you go to a mega church with millions of followers and they spew all sorts of bullshit there about evolutionary theory, and people follow. And the question is, go and read what evolutionary theory says. It doesn't say what they told you there. And I chose church and in the synagogue and in the mosque. Okay? So I think if you talk about civilization, I think if we can educate the public about this important concept, I think it's the greatest contribution to civilization. Because we keep talking about, I am coming from the Middle East, I know something about wars and hatred, and people keep talking about, let's get rid of WMDs. Religion and God are the biggest WMD. But that's all I can say.
Let's say more to, I mean, this talk is clearly an example of X plus CS that also Vivek mentioned in his talk. This is like how biology is related to computer science. And one of the things that as a computer scientist bothered me when I interact with the X is the lack of formalization. So if you take, for example, your definition of species, if you just answer the formal guys, so you now build a model that is based on some formal definition that you make that the biology side are bound to change them every now and then, not to mention other definition. How is the computer science that you interact with them?
You have to talk to biologists, you have to learn the biology. When I was teaching the bioinformatics course one time, I had a student from Moshe's group. I forgot her name now, but she was in the class and she did,
Kristen Roger.
I don't remember now, but she did the project and she gave a presentation at the end, and her conclusion about the project was, it's very hard to deal with biological data. She's coming, you do research in areas like formal verification and all of that, you assume that you know the input or what it is and stuff like that. In biology, there's a lot of uncertainty about everything along the step and along the way. And biology is not an exact science. It's not either this happens or not. Nothing does not happen. Nothing does not happen. I mean, this is one of the things about this evolutionary theory. All sorts of crazy things could happen. I mean, I don't want to start giving now outrageous examples. All sorts of things that can happen between human and chimp and gorilla. But the thing is that you have these notions of natural selection and you have notion of reproductive isolation and so on. It's not easy to come up with a model that captures all of biology. But what I would say is that if you want to work in this area, you can decide how much you want to go into the biology. Right? Some people stay very far from it. They write papers, very beautiful mathematical and algorithmic results. The only mention of biology is in the abstract. You know, here's where it would be relevant or useful. But in my opinion, if you want to have impact in biology, you have to get into biology. And one of the tough messages, lessons I learned in biology is that what we mean by modeling in computer science is not what they mean by modeling in biology. What we mean by modeling in computer science, for example, you hear people talk about state machines, for example. Here's a modeling technique, or Petri nets, here's a modeling technique. When biologists talk about model, they say, here's my system, I have written the set of differential equations for it. This is what they mean by modeling. There is no one system that you can model everything with it. Biology is so diverse, even across the human genome. Things are very, very diverse, and we don't know how they change and why they change like that. So I think if you want to do something that at the end of the day is useful in biology and used by biologists, there is no choice but to go there and focus on the biology and understand it. Any other questions?
So talking about the diversity, I think that's another aspect of more data, right? Because with more data, assuming they're from like evenly distributed sources, then it's more likely you get something that does not fit into your model. So will that be an indicator of something wrong with your model? And is there a way to use that kind of data to isolate them so that you can get a sense of what's missing there?
Yeah. Actually, the way we started doing this hybridization modeling is that we started by modeling this data set of yeast. And one of the interesting things is that if you take that model, the tree model, and you try to fit the data, that model is going to give you a distribution on the gene trees. We looked at the data did not fit that distribution. So now you start looking at this data and look at the outliers, why didn't they fit in that model? This is the notion of hypothesis testing, right? You have a null model, say, here's what I would expect under the null model. Then you can reject or accept, or reject or not reject the null model. If you reject the null model, you have to start looking for alternative. And that's where it gets interesting. Say, okay, it did not fit. I must have missed something in the model. And then you start looking at how can I extend the model so that the data fit. And of course you can, then after that, some other data is not going to fit and you keep extending. But yes, I mean, this is one of the ways we use it. It's not just about, this is how we detect hybridization. But usually when you have the model, one of the interesting things to me is that when things don't fit the model, because that's where there's something interesting to do. Right? Yeah. If everything fits the model, then we move on. But when things don't fit the model, that's where the nice science actually starts coming.
Definitely. So is there a good way of trying to figure out how that's different that could be helpful? For instance, you're saying like if a model does not match, you try to add something and to see if that's a better match. But is there a way to see from the error message that something could be the cause of this?
I think it's easier in areas than other areas. Here, like when we did not fit the model, it could have been gene duplication, it could have been hybridization. But this is where you need to talk to the biologist, because if I went to the biologist and said human chimp gorilla did not fit the model, the biologist will say, you can exclude hybridization, right? It could be probably duplication loss. We could have added duplication loss, something else did not fit well. So, I mean, the area of outlier detection and so on is an active area in machine learning. So this is not something about biology, because now we are looking at things don't fit within the model and you want to detect them. So there are two tasks there. First, finding what doesn't fit in the model. And second, trying to figure out how to augment the model so that it fits. I would say the first step can be done in a more systematic way. Finding what doesn't fit in the model. There are lots of tests. Hypothesis testing is actually part of that. Or you can go Bayesian and do all sorts of analysis that are not based on hypothesis testing. The second part, I would say that it requires some domain knowledge and domain expertise that you start saying, okay, I didn't fit. But you cannot now you can take a mathematical way, start saying, let me start adding parameters. But I don't take that. I prefer to take the approach of, no, let me try to understand the biology. Because I think that is the more intelligent way. The other one is the more brute force way. Add 15 more parameters, all of a sudden the data fits. Well, that would fit, that would work, right? It's by definition. Keep adding parameters, you will fit it. But the question is, why did it fit and which parameters you should add? And I would advocate the route of understanding the data rather than just brute force, add more parameters. There's beer. I don't know if you know that.
If you're a graduate student, faculty, or staff, you're welcome to join at Valhalla. We have a happy hour from 5, and we don't have upper bound, so.